Right, so chapter 44 is all about osmoregulation and excretion, how our kidneys work, and how we are able to maintain fluidity and keep in ions and other small molecules, and how we remove them when we get too many of them. So, our systems, whether you are human or any other sort of animal, have a fluid environment surrounding the cells. With the open circulatory systems, we have the hemolymph, and with the closed circulatory systems, we have the interstitial fluid and the blood. And homeostasis needs to be in order. We need to keep relatively constant concentrations of water and solutes to keep everything working correctly. Osmoregulation helps to regulate the solute concentrations and decide whether we need to lose or gain water. And excretion helps us to remove excess water along with metabolites that our body no longer needs and other waste products. So, osmoregulation again is based on this relationship between solutes. Cells have to be balanced in terms of their water input. We talked about this a lot when we talked about osmosis with isotonic and hypertonic and hypotonic cells. Osmolarity is focused on the solute concentration, and that's going to determine which way the water is going to move. If your solutions that you are looking at are isosmotic, then the water movement is going to be constant. Both locations, both solutions are fine. But if they differ in osmolarity, the one that is hypoosmotic, that has way more water in it than it has the solute, will have water move to the hyperosmotic, the one that has too little water. So there's an example. More solutes, water's going to move to the side with more solutes to try to balance things out, maintain homeostasis. There are osmoconformers and osmoregulators. Osmoconformers are going to apply pretty much just to your marine animals. They maintain isoosmotic conditions with their surroundings, so their internal osmolarity is not going to change. Osmoregulators will adjust their internal osmolarity to their water intake, or to allow water to be lost or water to be gained, depending on what type of osmotic environment they are in. They are able to do this using a concentration gradient, which requires a fair amount of energy, but this also allows them to live in many different habitats. A lot of animals are stenohaline. They can't deal with substantial changes in external osmolarity. Euryhaline animals can. This would be mostly your osmoregulators, but there are some osmoconformers that can deal with the osmotic shock more effectively, and they too could live in environments that have large ranges in this external osmolarity. Your marine animals, as we talked about, the marine invertebrates are osmoconformers. The marine vertebrates, and there are some invertebrates, are going to be osmoregulators. Marine bony fishes are actually hypoosmotic to seawater, and they, along with desert animals, will lose their water greatly as a result. They also gain salt either through diffusion or from their food. So, how do they make this work? They drink lots of seawater and they excrete salts to keep things in balance. While freshwater animals are in a hypoosmotic environment, so they are constantly taking water in. So they want to try to minimize how much water they're taking in and not lose solutes. So they do lose some salts from diffusion, and they help to keep the water balance under control by excreting lots of dilute urine. The salts get replaced by foods and they get taken up through their gills. So there's your marine fish and there's your freshwater fish. You'll notice that in the marine fish, they are taking in water from both seawater and ions from food. They are able to excrete some of those ions from their gills, and they're able to lose some of their water through the gills. They do not excrete a lot of urine because, again, they are losing water constantly to their environment. While in a freshwater fish, they are gaining lots and lots and lots of water. And so that's going to reduce their salt ions. So they take them up through their gills, they take some up through their food. And because they keep taking in water and they keep taking up ions, they lose both through lots of urine in their kidneys. And this is just a pretty wild one. There are aquatic invertebrates that might exist in ponds that aren't there for an extended period of time. And when that happens, they will lose pretty much all of their body water and survive in a dormant state. And then that is a tardigrade. And you can see the hydrated and dehydrated forms. And this adaptation is called, or adaptation is called anhydrobiosis. Land animals have undergone adaptations to be able to minimize water loss. The body coverings helped with this, they help prevent dehydration. Desert animals are able to get water or retain water through some anatomical features and also by living primarily at night. Um, they're able to maintain water balance, land animals in general, by eating moist food and producing water from cellular respiration. If you are an osmoregulator, you have to use up energy to be able to maintain the homeostasis within your cells, to maintain your osmotic gradients. How much energy you use up depends on the difference between the osmolarity of your surroundings compared to your cells, how efficiently or and how easily water and solutes are able to move through the animal surface, and how much energy is needed to move the solutes across your membrane. So there's a lot of factors at play in determining how much energy is going to play a role. Remember that you have your interstitial fluid or you have your hemolymph that bathes the cells, and you have to regulate the solute content that's present there so that your cells are taking up only what they need. Transport epithelia are cells that are specialized just for moving solutes in certain directions, kind of like we talked about with the arteries and the veins. They are arranged in these pretty wild tubular networks, and one example of them is in marine birds and their nasal glands, and it helps them to get rid of excess sodium chloride. Okay. So, section two, nitrogenous waste reflect both its phylogeny and its habitat. What type of waste it makes and how much can have pretty significant impacts on its water balance. And so the nitrogenous breakdown products from proteins and nucleic acids are probably the most significant waste that animals have to deal with. Um, Ammonia is pretty toxic, and as a result, some animals will convert it to less toxic compounds like urea or uric acid before they excrete it. Okay. So aquatic animals are going to leave it alone with ammonia. We see that mammals, some amphibians, sharks, and bony fishes are going to produce urea. And then reptiles, your birds, your insects, your land snails are going to make uric acid. So, why do we make different types? Again, toxicity, energy costs are going to play a role. The animals that are able to excrete nitrogenous wastes directly as ammonia need water, and that works great for your aquatic mammals. They can release it across their whole body surface or through the gills. Mammals, livers, and adult amphibians tend to convert it into ammonia into urea. And then the circulatory system takes the urea to the kidneys, where it's able to excrete it with less water than the ammonia would need. But this is an energetically expensive process, so you've got to have the energy to make it happen. We talked about the particular organisms that are going to make uric acid. It is pretty non toxic. It actually comes out more like a paste because it does not result in a lot of water being used up to do it. It does not dissolve in water, but it does require more energy. So, factors that are going to play a role in what wastes are excreted will depend on their evolutionary history, their habitat, aka how much water's around, the environment of the animal egg, and then keeping in mind that the amount of waste you have will influence directly the energy that the animal will need to expend. So, regardless of what type of animal you are, the excretory systems can have a range of setups. They are used again to regulate that solute movement between your internal fluids and the external environment that animals living in. Um, if we focus on urine, most are able to produce it by refining a filtrate from your body fluids. And there's four main functions that we see in our excretory systems: the filtration, where we filter those body fluids; reabsorption, where we take back, we reclaim solutes that are needed; secretion, where we add substances that are not essential, waste, non essential solutes from the body fluids to it; and then excreting, um, after the filtrate has been processed and has your nitrogenous waste, releasing that from the body. Okay. Um, Lots of tubules are typically involved. This is just a general setup of what you would see in excretory systems within an animal. So if we look at a protonephridia, a planaria, we see that protonephridiums are dead-end tubules which are connected to external openings, and they are able to excrete dilute fluids, so they help maintain osmolarity within the planaria. Metanephridia are found in earthworms. Um, each segment has one of these. They consist of tubules that collect coelomic fluid, and they are able to produce urine. Malpighian tubules are found in insects. They are able to remove nitrogenous waste from hemolymph and produce uric acid. And then we have our kidneys, the bean shaped, about 10 centimeter long organs that have lots of non segmented tubules that work with a dense capillary network. They are able to function in vertebrate excretion and osmoregulation. Blood enters via the renal artery and then it exits via the renal vein. From every heartbeat, approximately 20% of blood is pumped to your kidneys. Your kidneys have around 1,100 to 1,200 liters of blood flow through them daily, and the nephrons in the kidneys process 180 liters of filtrate each day, and then whatever is not excreted returns back to the blood. The urine leaves the kidneys through ureters and drains into bladders, which then leaves through the urethra. Okay, we're going to spend a lot more time on the structure of the kidney and the nephrons that make up the kidney. Nephrons are your kidney's functional unit. Each nephron will consist of a renal tubule and a renal corpuscle, which is just a bunch of capillaries that have a afferent arteriole that's coming from the renal artery, so where they get their blood. The renal corpuscle, again, is just a bunch of capillaries. It's kind of rounded. It's called a glomerulus, and it's covered by the Bowman's capsule. The capillaries, when they leave the glomerulus, can't say that word, save my life, form an efferent arteriole. And then there's this thing called a vasa recta, which is a capillary system, which you're going to see that basically surrounds and intermingles within the loop of Henle. And then from the capsule, we also have the renal tubule. Okay, so there are two main types of nephrons, and this is again where we separate all of those metabolites and the ions and water and other small molecules from our blood. We have cortical nephrons and we have juxtamedullary nephrons. The cortical nephrons make up the vast majority of the nephrons we find in our kidneys. The biggest difference between them, if you notice, is that the juxtamedullary nephrons have much longer loops of Henle, and so they are able to go further into the renal medulla. So you can see in this particular picture the vasa recta, you can see the arteries and the veins, you can see the Bowman's capsule, the glomerulus, um, And we're going to talk about the proximal tubule and the descending and ascending limb of the loop of Henle and the collecting duct as we continue on. So there's a stepwise process in which blood is filtered in the nephrons. It's going to move from the glomerulus into the lumen of the Bowman's capsule, and then it will move through the proximal tubule, the loop of Henle. There's a descending and ascending limb, a distal tubule, and then it will move down through the collecting duct so that it can be prepared for excretion. So we're going to look at each of those one at a time. Um, you have a cortex, which is found towards the outside of the nephron, and then you have your medulla, which has an inner and outer portion. The proximal tubule is found in the cortex. It changes not just the volume of your filtrate, but also its composition. The osmolarity of the blood filtrate that's present here stays relatively constant. Um, so we absorb ions and water, or sorry, in this tubule, ions and water and other nutrients are reabsorbed back into the blood or into the interstitial fluid and then taken away. And we also have toxic materials that are added to this filtrate. So things are becoming more concentrated, but we are not and we are losing both water and we are losing both ions, but the osmolarity is remaining relatively constant. And the descending limb of the loop of Henle is where we start to see some changes. This limb extends from the cortex into the outer and inner medulla. Only water is able to leave in this part of the loop of Henle. The aquaporin proteins that we've talked about before allow more reabsorption of water into that interstitial fluid. And that is able to be done passively because interstitial fluid is hyperosmotic to the filtrate, which is moving through the loop of Henle. So, all said and done, as it moves through, the filtrate is becoming more concentrated. There's more particles present compared to the water. When we move back up into the ascending limb of the loop of Henle, we're moving from the medulla to the cortex. It's at this point that the loop of Henle is permeable just to salt and other small molecules, but water has to stay put. So now we're working against the osmolarity of the interstitial fluid. There are two regions for this loop of Henle. The thin region is where salt moves passively. The thick is where energy has to be expended because we're using a concentration gradient to move NaCl out. So as we do this, our filtrate is becoming more dilute since we're losing the ions that are present in it. Within the distal tubule, the osmolarity of the filtrate remains pretty constant. There's both secretion and reabsorption occurring, but it's pretty specific as to what takes place. This is in the cortex. We have potassium and hydrogen ions that are secreted into the filtrate. And then we have salts and bicarbonate ions that are reabsorbed. And some water is passively recaptured as well, both in the proximal and in the distal. The ion movements help to kind of help with pH regulation. The collecting duct takes what we have now of our filtrate and helps to move it from the cortex through the medulla to the renal pelvis. It takes advantage of the transport epithelium that are present in the nephron to help to process the filtrate into urine. It's able to reabsorb any remaining solutes, and it will produce a urine that is hyperosmotic to your body fluids. Depending on your body's needs with volume and pressure, it can also play a role as to how dilute or how concentrated that urine is. So here is a picture step by step of what's kind of taking place. So you can see the red arrows are showing you active transport, the blue arrows are showing you passive transport. We talked about how both water and ions and molecules can leave in the proximal tubule. In the descending loop of Henle, only water is able to leave passively. In the ascending loop of Henle, we see that salt is able to leave both passively and actively. In the distal tubule, we have selective ions and molecules being able to move in and out of the filtrate. And then in the collecting duct, we have salt leaving actively, we have water leaving passively, and we will talk a little bit about how some urea will also leave. And what determines what's going to be left in your blood? It's controlled by three processes: filtration, secretion, reabsorption. Through filtration, the fluid is forced by blood pressure from the glomerulus into the lumen. In secretion, the filtrate gets joined by substances in the interstitial fluid, like plasma. And then reabsorption is taking back materials that can be used by the body, small molecules, sugars, vitamins, water, and organic nutrients, things the body needs at that particular time. Um, Being able to conserve water is a very important terrestrial adaptation since you're not living in the water. We are producing hyperosmotic urine, and we're able to do this because of the energy the body decides to use towards this to transport solutes against their concentration gradients. The two solutes that play the biggest role in determining the osmolarity are your salt and urea. So we talked about the two solute models looking at what's happening with those two particular substances. We talked about how in the proximal tubule, the osmolarity remains roughly the same because you're losing both salt and water. And the descending, because you're losing water, your osmolarity is going to increase pretty substantially up until it's always going to be basically continue to lose water until it reaches the osmolarity of the surrounding interstitial fluid. In the ascending loop of Henle, we are losing salt since we're not losing water to reduce that osmolarity. And then the distal tubule, we talked about how it remains constant. And then the collecting duct is going to vary depending on what the needs are of the body. Why are we able to do this? This is a countercurrent multiplier system with the loop of Henle that helps to keep the salt concentration in the kidney's interior pretty high. We talked about those vasa recta vessels, blood's moving in opposite directions because in theory, this wouldn't work because you've got, again, everything wants to reach osmosis. So how the heck are you able to get the kidney and specifically the loop of Henle that its needs and not have water move into the kidney's interior? So the vasa recta veins are able, or the capillaries are able to get the kidney its nutrients and don't interfere with the gradient specifically. It just allows the filtrate to use active transport to move the salt against its concentration gradient. So in the collecting duct, salt's not going to be able to leave, but only what water can. So this will help to generate either dilute or concentrated filtrate. We do see some urea leave. This also helps with the osmolarity as it's adding to particles that are present in that interstitial fluid. It does get recycled via the loop of Henle, but because there's constant leakage, there's always some urea there to help with that interstitial concentration. So both the urea and the salt are able to make it possible for the kidney to make urine that is hyperosmotic to your blood and not have it take water from the blood or from the interstitial fluid elsewhere in the body, but it's actually isoosmotic to the inner medulla's interstitial fluid. So the kidney's osmolarity is going to be hyperosmotic to pretty much anywhere else fluid-wise in the body. So this is kind of showing you what's happening with the osmolarity values. I talked about them a little bit. You can see that starting out around 300 milliosmolar, and then as it continues to go down the descending loop of Henle, it's matching up with the interstitial fluid osmolarity on the far right. As it moves back up through ascending and it starts to lose salt, the osmolarity diminishes. And then as it goes down the collecting tube, the water and the salt and the urea will leave until the osmolarity of the urine, the filtrate, the processed filtrate, is going to be isoosmotic with the interstitial fluid within the kidney. So nephrons are going to vary in different vertebrate classes based on what the osmoregulation needs are for that particular habitat of the animal. So in mammals, we talked about how there were two types of nephrons, the cortical and the juxtamedullary. That's basically how we're able to conserve water. The juxtamedullary nephron goes deep down into the medulla, so it's going to cause an even steeper concentration gradient. So it's going to be able to produce urine that is more hyperosmotic to your body fluids. When you live in a dry environment, you're going to have longer loops of Henle to help have those osmotic gradients that are pretty steep. While if you're in fresh water, so you got plenty of water available, you aren't going to need as steep of an osmotic gradient. You're not going to need to use up as much energy to produce that urine. And so your loops are going to be fairly short. Birds and reptiles also have shorter loops of Henle, excuse me. So you would think that they would have dilute urine, but again, they conserve their water by generating uric acid. So they put their energy into making that form of um, of making those changes to ammonia to produce the uric acid. There's some other reptiles that have only cortical nephrons, but they are the ones that excrete their waste as uric acid. Freshwater fishes, amphibians. Freshwater fishes want to hold on to their salt in their distal tubules, so they are able to excrete large amounts of dilute urine via cilia. And then the amphibians have a similar kidney function, but they conserve water when they are on land by taking in water or reabsorbing water from their bladders. Marine bony fishes are going to be hypoosmotic compared with their environment. So they're going to be the ones that are losing water. They have small glomeruli in their kidneys. Some of them don't have them at all. So they don't filter a whole lot. They want to hold on to as much water as they can. They don't make a lot of urine. What their kidneys are able to do is to help get rid of the ions that are found by them drinking the seawater. Those ions are secreted into the proximal tubules and then excreted through the urine. Hormones can play a role in how your kidneys are functioning, maintaining water balance, and dealing with blood pressure changes. So, this bat can produce both dilute and concentrated urine. Depending on what type of urine it's producing, it's able to both reduce its body weight pretty quickly or ingest large amounts of protein and still able to meet its water needs. So the urine's osmolarity is regulated by both hormones and by the nervous system. ADH, also known as vasopressin, antidiuretic hormone, makes the collecting duct epithelium more willing to transport water into that interstitial fluid. So if the blood osmolarity, not the kidney osmolarity, not the kidney interstitial fluid osmolarity, but if the blood osmolarity increases, um, ADH will be released. This helps to reabsorb water. So that's going to make your urine more concentrated. And eventually, as that water moves into the blood, is able to reduce the osmolarity of your blood. When ADH binds to receptor molecules on the collecting duct epithelial cells, it increases the number of aquaporin proteins, which is what allows the water to move through. If there's an ADH mutation, this can cause severe dehydration. You're not able to bring the water back in, and it can cause diabetes insipidus. Alcohol is considered to be a diuretic because it prevents ADH from being released. So, again, why would you have an increase in blood osmolarity? Maybe you went for a 5-mile run and you were sweating a ton. And so, as a result, your blood has lost water, and we need to take water back in. Osmoreceptors in the hypothalamus are going to cause ADH to be released from your pituitary gland. Your collecting duct, that final step in your nephrons, is going to allow water to move across it so that it can be moved into the interstitial fluid and be reabsorbed. As a result of being reabsorbed, it's going to help to readjust your blood osmolarity. Another more physical indicator of the dehydration is that you are thirsty. So you can ingest water, which will also help to reduce the blood osmolarity and get things back to a homeostatic level. Okay, so going back to cell signaling, the ADH hormone finds its receptor on the collecting duct cells. It sends off that second messenger, CAMP, which causes um, vesicles to be formed that are from taking in water from the collecting duct, and then they are able to move through those channels to allow to water to go back into the interstitial fluid of your kidney. RAS, renin, angiotensin, aldosterone. This also plays a role in homeostasis. The one we just talked about with ADH is only dealing with the osmolarity of your blood. The glomerulus has a way to recognize blood pressure changes. And when there is a drop, it will allow the juxtaglomerular apparatus, JGA, to release renin. And renin will lead to the formation of angiotensin 2. Angiotensin 2 actually causes a physical change in your arterioles. It constricts them. So it causes the blood pressure to go up. And as the blood pressure goes up, the blood flow to the kidney is reduced. And the reduction in that blood flow will cause aldosterone to be released. Aldosterone acts on the distal tubules of your nephrons and your collecting duct to take in more ions and more water. That's going to help to increase your blood volume and, as a result, your blood pressure. So, again, the first initial change. So, why would this happen? Again, you've lost water from exercise, dehydration, you haven't had enough water, or you've had some sort of injury that's resulted in blood loss, so the blood pressure has dropped. So when that is recognized, the juxtaglomerular apparatus releases renin. Angiotensinogen, a precursor to angiotensin 1 and 2, is released in the liver or is present from the liver. Renin acts on that to turn it into angiotensin 1. ACE, another enzyme, acts on it to turn it into angiotensin 2. Angiotensin 2 on its own is able to constrict those arterioles, which physically causes a change in blood pressure, Boyle's law. And then angiotensin 2 is also chemically able to encourage the adrenal gland to release aldosterone. That particular chemical absorbs both water and sodium, so that's going to cause your osmolarity to stay fairly constant, but it's going to increase your blood volume. And as you increase your blood volume, that's going to help get your pressure back up too. So the decrease in blood pressure that's recognized at the by the juxtaglomerular apparatus is able to result in both a physical change with angiotensin 2 in terms of constricting your arterioles and a chemical change with the production of aldosterone. So both ADH and RAS will cause water reabsorption to take place. But remember, ADH is only dealing with osmolarity changes. RAS is dealing with specific changes in blood volume. ANP is able, because we got to have the counterbalance, the negative feedback will oppose RAS, and it's going to be replaced so that we don't continue to take in water more than we need to. ANP is going to be released in response to blood volume increasing and as a result of blood pressure increasing, and will stop renin from being released further.